talk today is by Jonathan Ma, and from the Lohmiller and Garud labs.
Okay, I think this is the last talk before the fine folks at QBio Edge, so let's have a little bit of fun with it, okay? How's it going, guys, gals, and non-binary pals? My name is Jonathan Ma. I'm a bioinformatics PhD student co-advised by Professor Kirk Lohmiller and Professor Nadita Garud. And time permitted, today I'll be discussing some of my hashtag soon-to-be submitted work concerning inference of demography and selection in the human gut microbiome. But before we get there, I'd like to start off with a quick story about, I know we're all thinking it, that's right, you guessed it, the US National Census. So the way this works is every 10 years, the government sends a bunch of people with clipboards to ask questions such as how many people are living, what kind of people are they, and where are they living? And this information is used to distribute voting power in the House of Representatives. So to some extent, it does have meaningful impact on our daily lives. As an example, let's consider Los Angeles. One of the things that the census tracks is how population sizes change over time. Here we can see that as of the year 2000, Los Angeles had an estimated population size of about 3.7 million people, increasing to 3.8 and 3.9 million in the following decades. And if you're like me and politics fills you with dread, then you might want to know why. There's plenty of reasons why more people want to live in Los Angeles. We have beautiful weather, we have great food, and one of our baseball teams, the Dodgers, even won the World Series in the year 2020. However, in tracking the census, we also know that relative to the year 2020, as of 2022, Los Angeles has instead seen a decrease in estimated population size. And if you're like me and politics fills you with dread, then you might want to know why. There are plenty of reasons why fewer people are living in Los Angeles. For example, the COVID-19 pandemic, the high cost of living, and our other baseball team, the Angels. Altogether, the census gives us important insights into demographic trends, which have very real, very tangible effects in our lives. What I do is very similar, except instead of asking how many people live in Los Angeles, I ask how many bacteria live in people. And if you're like me and bacteria fills you with bacteria, then you might want to know why. Our system of study is the bacteria that live in, on, and around us, otherwise known as the microbiome. In particular, I study the bacteria present in our guts. Here is a simple cartoon schematic of a person and their bacteria, with these lines on the right-hand side representative of bacterial genomes. Let's consider, for one example, a single species, Rheumatococcus bromine. You might notice that not all of these lines are the same color. This is to represent the observation that typically there are between 1 and 4 strains of each species per host. That is to say that the strains present in my body are completely different from the strains present in your body. In summary, the human gut microbiome presents an extremely interesting and complicated model system in which we can ask fundamental basic science questions about evolution and ecology. Thus, we began our investigation by asking how nucleotide diversity might differ by geography. There exists a fairly substantial body of work suggesting that the microbiomes of people living in industrialized or urbanized societies, such as everyone here, have seen general reductions in microbiome diversity at the level of species, gene, and nucleotide diversity. This is important, particularly in the context of human health, because such depletions in diversity are associated with negative or deleterious health outcomes, such as increased susceptibility to infectious, chronic, and developmental diseases. Here's how we approach this. We used fecal shotgun metagenomic sequencing data to investigate population genetics questions in the human gut microbiome. More informally, instead of calling this work population genetics, we like to call it population genetics. We computed the nucleotide diversity both within hosts and between hosts for several bacterial species present in at least one of 2 cohorts. One from a rural African population and another from an urbanized North American population. Let's take a closer look at the data. Hey, it's our old friend, Rheumatococcus bromine. I've separated the data by cohort, with the African gut microbiome shown in blue and the North American gut microbiome shown in yellow. Each smaller circle represents the within-host nucleotide diversity of a single person, with a box and whiskers representing the distribution. Separately, I show larger diamonds, also color-coded and typically found above the distribution of smaller circles. This represents the average across-host nucleotide diversity computed in a pairwise fashion. We've done this for all species which are present in at least 5 hosts in both cohorts. Quite interestingly, by means of an unpaired 2-sided Wilcox and Rank sum test, the difference between the distribution of African and North American within host diversity is statistically significant in 9 out of 13 species after controlling for false discovery rate. Here on the right, we show the mean across-host nucleotide diversity of all species present in either cohort, irrespective of overlap. And again, quite interestingly, we find a statistically significant depletion in nucleotide diversity found in North American gut microbiomes relative to African gut microbiomes. This was also done via a two-sided Wilcoxon ring subtest. Next, we turned our attention towards quantifying how many bacteria live in people. Given our findings of depleted nucleotide diversity, we focused the remainder of our analysis on North American gut microbiomes. We're interested in gut microbiome diversity across human populations, so to garner a representative data, we constrained our analysis to only a single strain of each species per host. We leveraged a summary statistic of genetic variation known as the site frequency spectrum, which we can visualize here as a histogram, by which the x-axis tells us how rare or common a mutation is, and the y-axis denotes the proportion of mutations which fall into each level of rarity. As an example for how to read this first bar on the left refers to singletons, which are mutations which only manifest once in a population. Furthermore, we can split this purple bipartisan SFS into putatively neutral synonymous mutations shown here in red and assumed negatively selected non-synonymous mutations shown here in blue. The SFS is known to be subject to three major classes of forces, being demography, linkage, and selection. However, remember, we're working with bacteria, which have an extremely high rate of recombination. Thus, to some extent, we can ignore linkage, meaning that our synonymous red SFS is almost entirely subject to the evolutionary force of demography, which we can infer. We use a statistical inference package known as DADDY to infer demographic models from the synonymous SFS. On the right here, I show several different cartoon schematics of different model specifications. The width of each schematic corresponds to the effective population size at that time. Let's proceed by walking through an example. Here I show the synonymous site frequency spectrum of our old friend Ruminococcus bromei. In darker colors, I show the empirical data, while in lighter colors, I show the spectrum simulated under our best fit demographic model. Speaking of demographic models, the way that we fit these is by exploring a two-parameter likelihood surface using a gradient descent-based approach. Our two parameters are nu, shown here along the x-axis, which represents a ratio of current to ancestral population size, and tau, shown here on the y-axis, which represents the amount of time which passes in an epoch given in units of generations per 2 times the ancestral population size. A difference of approximately 3 log likelihoods corresponds to an approximate 95% confidence interval. And in summary, the model parameters, which simulate a model spectrum most closely fitting the observed data, are assumed to represent the underlying evolutionary history. Want to see me do it again? We did, for 27 bacterial species simultaneously across the human gut microbiome. Here on the right, I'm showing a summarizing scatter plot where the x-axis is identical to that of the likelihood surface. The y-axis has been converted to actual years, actual units of time in years. I'd like to highlight this dotted red line on both the likelihood surface of the scatter plot, which separates the parameter space between contractions and expansions. We find that bacteria are inferred to have varying demographic histories. I'd like to note that this time frame of around 10,000 years is quite meaningful in the context of human history. A similar study by Omar Cornejo et al. Found a population demographic expansion in the cavity-causing oral pathogen Streptococcus mutans on a timescale roughly coincident with the onset of human agriculture 10,000 to 20,000 years ago, in part due to increased ecological niche availability as a consequence of a shifted diet. We suspect that perhaps something similar is happening in Ruminococcus bromei, which also sees an expansion on a timescale roughly coincident with human agriculturalization, and has been shown to increase in abundance in the presence of diets high in certain starches, a hallmark of urbanization. We don't quite have enough time to walk through each and every single one of these species. However, I personally find it incredibly interesting and compelling how, despite cohabitating in the same physiological environment, each of these species has their own distinct evolutionary history. But demography is only one part of the story. Next, we asked how selection might act differently across the human gut microbiome. We used a statistical inference package known as FitDaddy. It's kind of like Daddy, but fit. The major difference here is that FitDaddy instead leverages this blue, non-synonymous site frequency spectrum to infer selection conditional on demography by treating the putatively neutral red synonymous sites as a reference for neutral evolution. Quite nicely, when we condition on demography and have high enough recombination, then the unaccounted for primary evolutionary force acting on non-synonymous mutations is selection, which we can infer. Here I show the distribution of fitness effects, otherwise known as the DFE. This is a fundamental entity in genetics, which describes the proportion of variants and to what extent selection acts on them. We can visualize this as a histogram in which the y-axis describes the proportion of sites, and the x-axis denotes whether mutations are more neutral or more deleterious. I've also color-coded the bars to correspond with lethality, with darker reds indicating more highly deleterious mutations, while lighter grays correspond to more neutral mutations. Want to see me do it again? We did, for 27 bacterial species simultaneously across the human gut microbiome. Here I show a phylogenetically sorted panel of the DFE for all of these species. I'd like to draw your eye towards some broad trends across genera. The Eubacterium genus, here highlighted on the bottom, tends to have extremely deleterious DFEs. By contrast, the Bacteroides genus, highlighted here in the middle, tends more towards weakly deleterious or moderately deleterious DFEs, providing some evidence of phylogenetic congruence of the DFE. However, overall, hopefully as you can see, the story of the DFE is similar to that of demography. Each bacterial species has its own distinct evolutionary history. Having found that the DFE varies across species, next we asked whether the DFE might vary within species. Let's revisit this cartoon from earlier. All of the analysis I've talked about so far has been over what's known as core genes, that is to say genes present in at least 95% of samples. However, the microbiome also has accessory genes, which we define as genes present in between 30 and 70% of samples. Quite importantly, accessory genes are known to encode for highly specific functions, such as antibiotic resistance, toxicity, and even specialized metabolic pathways for digestion. We did this whole song and dance again. We computed the site frequency spectrum. We split it into synonymous and non-synonymous mutations. We inferred demography. We found the distribution of fitness effects, but this time for accessory genes. And quite interestingly, in the species Bacteroides theta iota omicron, we found that core genes have a much more deleterious DFE than accessory genes. I'd like to highlight that this x-axis is on log 10 scale. So the selection coefficients acting on core genes are several magnitudes more deleterious than those acting on accessory genes. Want to see me do it again? We did for 7 species across the human gut microbiome. Due to limitations of data paucity, we were not able to perform this analysis for all 27. However, of these 7, the difference in the DFE between core and accessory genes was found to be statistically significant in 5 of them, when considering a null expectation incorporating effective population size, potentially suggestive of not just differences in selection, but also differences in the underlying population structure between core and accessory genes. Overall, we find a general trend across species in which core genes are subject to more deleterious selection than accessory genes. In summary, our work recapitulates the observed reduction of genetic diversity in North American gut bacteria. This study is the first known instance to leverage the site frequency spectrum to infer demography and selection in such a large number of species, which is particularly useful in elucidating the prokaryotic branch of the tree of life. And even within species, we found that different parts of the genome are subject to different levels of selection. A limitation of our approach is that our method of inferring the DFE can be considered as taking the average across hosts. However, in the future, finer scale inference on a per host basis could reveal exciting insights into personalized medicine. I'd like to acknowledge my many, many wonderful colleagues. Shown here on the left is the Lormuller Lab, and shown here on the right is the Groot Lab. All of my colleagues are awesome, but some of them are awesome. And here, I've put a little gold star next to people who are presenting today. I encourage you to seek them out and talk to them during the poster session. And lastly, this work was supported in part by funding from the Systems and Integrative Biology T32 training grant. Oops. I'd like to thank you for your time and attention, and otherwise I'd be happy to take any questions if there are any, and if I don't get to you, please feel free to reach out to me at this contact information.
Any questions for Donovan? Yeah. Donovan, could the accessory genes that you're appearing share quite a different demographic history, something that's pretty close, and also compared to something as well. So how do you leverage like doing this analysis across all accessory genes, given that the demographic history is quite very different?
That's a great question. I'm going to repeat it for the microphone. So the question is whether or not accessory genes may or may not have a different demographic history than core genes. So that's kind of a complicated question. So in truth, there's actually a lot of quality control that we have to implement into our SFSs to make sure that they're interpretable for our model-based inference. Here I'm showing 3 examples of an SFS on the top without any form of clade control. In the middle, I'm showing what happens when we do control for the largest clade. And on the bottom, I'm showing what actually the data we're working with. So a key sort of limitation of our approach is that we have to take only data from the most prominent strain of each bacteria per host. And as a consequence for that, our assumption of population structure is based on core genes. So you're correct in that if accessory genes have a different underlying population structure than core genes, then our inference may not necessarily be accurate, but that's kind of just a consequence of our approach.
Sure. Okay. So that was really cool. I don't know that I understand, but it strikes me that there are two kinds of demographics happening, right? There's the demographics of the bacteria, and your time scales are older 10,000 years or so. So there's also lots of demographic changes in the human population.
That is true.
At that time. So I'm wondering if, like, as you said, this is kind of an average. In a way, it's kind of treating the humans as kind of like a mean field, like a mean, or maybe I'm wrong about that. Where I'm going with this is, to what extent can you learn about how this is happening in tandem with the human demographic changes? Or is that making sense even when I'm asking? In the sense that, like, population size of humans are increasing, there are potentially correlations in different populations between their gut microbiota, right? Like there might be similar, like a kind of variance of that. And I'm just curious if that can enter into this in any way, or if you could sort of modify the model to inform both of those things at once.
Yes, so I'm going to sort of re-summarize the question if that's okay.
Yeah.
So Professor Eric Gates is asking, in the context of human demography, might that be impacting bacteria, for example? So on a timescale of roughly maybe 100,000 years ago is when we suspect that out of Africa, migration to have occurred. The question is, does that affect our demographic inferences? I would say yes and no. So if you recall, the bulk of this analysis was performed over North American gut microbiomes. If we were to have sufficient data to compare the demographic differences of gut microbiota in both North American and African microbiomes, then I would say we might be able to pick up that sort of signal from 100,000 years ago. But in the case of...
I noticed that 100,000 years on the fly.
That is
so
true.
It seems like maybe there might be something there. That's, yeah.
That sounds like an exciting avenue for future investigation.
Okay.
Yes.
One more question. I was related to the previous question. I was wondering if you could give kind of an idea for scale required to differentiate between different microbiomes. It's interesting if you have this graph out in front of a bacteria and have a line of history. Cool. But you and I have different strains of bacteria. I adopt say my microbiome significantly are different.
I'm going to repeat that question while I scroll to the slide. So the question is, how can we quantify the scale of differences between microbiomes? Is that accurate?
Right. Like, at what scale could you? Like, you were looking at from America versus South Africa, but if you quantify the difference, is it possible to quantify the difference between West and France? Would you have to scale that up a lot? And what rates is it possible to quantify difference?
That's a very interesting question. I do not have any insight into that. The only thing I'd really be able to offer is to do the experiment. Part of what makes this work so exciting is that this is the first try at inferring demography and the DFE in bacteria. So in the future, people could definitely try and more finely tune what the differences are between very closely related populations, but that requires, first of all, the data to do that, and then the analysis and stuff. So I don't have any immediate insights towards answering that question, but we can quantify differences between DFEs. So here I'm showing on the top right is a likelihood ratio test of parameters. The DFE is assumed to be gamma distributed, meaning that it's parameterized by an alpha and a beta. And we can test a constrained model in which we assume that two sets of genes have the same DFE versus the denominator, which is when two different sets of genes have independently fit DFEs. And then this LRT statistic lambda multiplied by 2 asymptotically is chi-squared distributed, from which we can form confidence intervals and then more definitively say this DFE is different from this other DFE. But more fine-tuning based on geography does require a lot more. Great question, though.
Great, that was wonderful. I think I also want to thank you for this very compelling demonstration of how effective it is when you are practicing your science communication.
And on that topic of practicing science communication, I'd like to invite the